I think Sati, you can utilize this time in introducing
Yes,
Dr.
sir.
himself.
All right. So once again, sir, thank you very much for this highly informative session. And now it is my pleasure to introduce Dr. Hifzur Rahman Siddiqui, a senior assistant professor at Aligarh Muslim University and program director of the Molecular Cancer Genetics and Translational Research Program. He earned his PhD from the Indian Institute of Toxicology Research and completed postdoctoral training at leading universities in the USA. Dr. Siddiqui's research focuses on cancer biology, pharmacology, and chemoresistance. He has published over 75 research articles in high-impact journals and is recipient of several prestigious honors, including the SPUR Young Scientist Travel Award by USA, IABS Outstanding Cancer Research Award, and the SPER Innovative Researcher Award. We warmly welcome Dr. Siddiqui to deliver his talk on epigenetics and cancer. Thank you very much, sir, for joining us. Over to you.
Thank you, Swati. Thank you very much. And I am extremely sorry because due to the maybe there is a temporal epigenetic changes of my tone. So before going to start, where is the window? Yes. Is it visible now?
Yes, sir, it is.
Is it visible?
Yes, sir.
Okay. Thank you. Thank you very much. It is not expanding. Yes. So the topic is epigenetics and cancer. So this is a hot topic, and this is the recent topic. Most of the scientists working, the number of scientists working on this particular field. Now, before going to talk on the epigenetics part, and I am giving you, means you people are listening so many things on cancer, precision medicine. So I'm just giving you a few sentences on cancer. So this is the scenario of cancer. It is now it's 18 means maybe it is around 20 millions, and into by 2040 the statistics would be almost 30 millions. Means maybe the difference is almost 12 million. So means and the number of things if you change particularly our lifestyles means we can prevent almost, according to the USA American Cancer Society, we can prevent almost 42% cancers. In Indian case, according to the ICMR, we can prevent almost 60% cancer just by changing our lifestyles. So this is the means a few line on cancer, and now the main topic, epigenetics. Now the question is, what is epigenetics? The Greek word epi, which means above or on the top, and that is part of the genetics. So the above or the changes which is above the genetics. Genetics means this is the we know what is the simple definition of genetic: inheritance and variation, and the key molecule is the DNA. Now the epigenetics refers to the study of how genes are turned on and turned off without altering the DNA sequences. So without changing our DNA genetic compositions, how the gene expression is turned on or turned off, or how this gene expression is changes during our life, or during the disease conditions, or during our different lifestyle conditions. Now this term epigenetics, which was coined by C.H. Waddington, you can see. So he is the scientist who coined the term epigenetics. Now you can see from the right pictures, there is a book. So if you consider the book is our genetics, the marker, this here book marker, they are the epigenetics. So the content is same. The content is same all the persons or the same persons, or maybe you can consider the for genetic purpose you can compare the twins. Okay, I will show you some pictures. You can compare the twins, monozygotic twins. They have 100% similar genetic compositions, but the bookmarks or the epigenetic marks, they are different from person to person. Even in monozygotic twins, those have the 100% genetic similarity. Now this molecular modification of chromatin, some of these modifications are here, but we'll discuss later in details. So before going to this, I will show you a 2 to 3 minutes video. This is the summary of epigenetics.
There are over 100,000 skin cells in 1 square centimeter of skin. Each of these cells contain close to 2 meters of DNA, which contain our genetic information. All the genetic material in our body is called the genome. Because our DNA is very long, cells package up their DNA together with proteins known as histones. The DNA and protein complex is known as chromatin. DNA is wrapped around histone proteins, forming repeated units of nucleosomes that look like beads on a string. Chromatin is condensed further to form a chromosome. We have 46 chromosomes in total, stored in the nucleus. The core of one nucleosome is composed of DNA wrapped around a histone octamer that consists of 2 copies of the major types of histones, H2A, H2B, H3, and H4. This organized DNA protein complex allows the cells to regulate what genes are expressed and when. All the cells in our body contain the same DNA sequence. Skin cells, muscle cells, and liver cells contain the same DNA sequence. Yet all these cells have different structures and functions. The reason is that different cells only use or express certain genes. Within each cell, the DNA and also the histones can be tagged by tiny chemicals that modify gene expression. These chemical tags cause some genes to be turned on and some genes turned off. For example, muscle cells will have genes turned on that a muscle cell requires, and will turn off genes that a liver cell requires. Similarly, the liver cell will have genes for muscles turned off and genes that a liver requires turned on. These modifications are known as epigenetic modifications. Epi is Greek for above the genes, so modifications above the genes or on the genes. Epigenetic modification brings about lasting changes in gene expression. Let's have a closer look. Here is a normal human cell, and we are pulling out its genetic material. The DNA is wrapped around nucleosomes, which are made up of histone proteins. Coming off the histone proteins, we have histone tails. DNA is made up of a combination of 4 nucleotides, G for guanine, C for cytosine, A for adenine, and T for thymine. There are 2 main epigenetic modifications. The first one we will talk about is DNA methylation, where a methyl group is added directly to a cytosine residue that exists in a cytosine guanine sequence, or CpG sequence for short. So for example, here we have many CpG sites that make up a CpG islet. And here the cytosines are methylated. The methylation of CpG sites in promoter regions is associated with gene silencing. The second type of epigenetic modification is histone modification. This is where acetyl or methyl groups are directly added onto histone tails, and this will modify gene expression. For example, histone 3K9 acetylation correlates with transcription activation, and histone 3K27 trimethylation with transcription repression. Genome-wide patterns of DNA and histone modifications, or epigenome, are established during early development and are maintained during cell division. In cancer, these patterns are altered and disrupted.
So this is just a short video, just to summarize what is epigenetics and how it's related to our DNA and histone proteins, the proteins which wrap the DNA. Now here you can see this is the monozygotic twins, and beautifully you can see, though they have the 100% genetic compositions are same, 100% monozygotic twins have the 100% compositions are same, but you can observe their hair color is different. And why this hair color is different? This is the questions. Here you can see this is the same dog. Okay, the 2 eye color is different. So this is here it comes through the epigenetic regulations. Now, how this epigenetic in or genetic compositions as well as epigenetic influence our whole phenotypes. So these epigenetic influences can be influenced by, you can see, microbiota. The microbiota, this microbiota present, the millions of microbiota present in our mouth cavity, as well as in our lungs, as well as in gut. Then also these epigenetic changes are also influenced by diet. Suppose this epigenetic whole composition is, you can find from, suppose, methionine rich food. If someone take methionine rich food, they have a different epigenetic composition, and then maybe they are siblings, or maybe they are twin sisters or brother. So diet also influence this one, because it means here it has been already reported that suppose those who are non-vegetarian and those who are vegetarian, they have different epigenetic compositions, as well as some means in race wise. Suppose if some Brahmins, because they are mostly dependent on vegetarian food, their epigenetic composition is quite different from the Rajput or Patan. So now this is, here you can see another is a disease exposure. Some disease also, some bacterial or some viral also influence this disease epigenetic compositions or epigenome. We will discuss the details about the epigenome, what is epigenome. Then psychological state, pollutants, lifestyle, toxin, chemicals also influence your epigenetic influence, and then exercise or physical activities. So means this combination of our genetic compositions as well as the epigenetic or epigenome combined together to form our whole human phenotype. So here this picture I have taken from the NIH website, and here you can see how this epigenetic mechanism, it starts from the uterus to the childhood to the adulthood, and from the aging up to the death. So here you can see these changes. Sometimes it is a permanent, sometimes it is a transient, mostly it is transient. So here you can see this epigenetic mechanism. They are developmental, environmental, chemicals, drug, pharmaceuticals, aging or diet, and how they in already you have shown the whole picture in the video. So this is the histone tail, and this is the target for the epigenetic, mostly target for the epigenetic modifications other than the DNA sequences. So as you studied during the undergraduate or postgraduate molecular biology, here you can see there are two types of chromatin in our chromosomes. One is euchromatin. What is the meaning of euchromatin? Eu means true. This is the true chromatin. True chromatin, why it is called true chromatin? Because they control the gene expressions. And another part of the chromatin is heterochromatin. Means there is a famous chromosome, you have studied the polygyny chromosomes or Lembrash chromosome. In polygyny chromosome, here you can start with the band and interband. So this interband part is the euchromatin, and the band part is heterochromatin. And why it is a heterochromatin? Here you can see these heterochromatin part or band part, they are highly compact. Why they are highly compact? Because of the high level of methylations. And this methylation causes the compact nature of this heterochromatin. And euchromatin, mostly here you can see this less methylations, more acetylations, and this acetylation making accessible to different proteins, and that's why proteins like RNA pol or DNA pol, RNA polymerase or transcription factors and other things, and so they can express easily. So this is means within the chromosome, you can see this is the difference between how these epigenetic changes regulate the gene expressions. In euchromatin or true chromatin, where the gene are highly expressed, or they are active part of the chromosomes, and heterochromatin, they are the inactive part of the chromosomes. Now here you can see this. Suppose this is a person A and person B. You can see this is the white blood cells, or you can take any cells. Okay, what is it? The hepatocytes or skin cells or pancreatic beta cells, gamma cell, whatever it is. So you can take the two cells from the person A, B from the similar type. Here the example is white blood cells. Both the person have the same DNA compositions. So DNA is same, nucleotide sequence are same, their genetics are same. But when this epigenetics changes, here you can see here the epigenetic is different from two persons, person A and person B. Here you can see 100% methylation is there, but here it is 50% methylations. Now, depending on this methylation, maybe the gene will is going to be a turn. Some genes are going to be a turn on, or some will turn off. So currently, here you can see some examples. This is the recent paper in Nature Cancer. So there are so many medicine targeting the epigenetic targets. Here you can see there are so many medicine already there. Some are in the under clinical trials, phase 1, phase 2, or phase 3, or some are already in the clinics. few are in the clinics. Here you can see this is the some of these drugs and how they target different epigenetic modifiers. Or there are three types actually: modifier, writer, and recognizer. So we'll discuss in later in our slides. And these are the other than the cancer in other field also you can see. So here you can see these are suppose this blue color, they are the neurological modifier, okay, epigenetic targets of neurological diseases. So some are the in yellow, they are the viral infections, and this green color here you can see both. This green color is immunological, but it is also used in cancer. So next is another beautiful examples we all have studied during our BSc classes. This is how this environment influence the epigenetic changes. Or here you can see when a mouse take the normal diet. So this is the normal diet they produce. There are so lots of yellow color plus obesity prone diseases, obesity mouse. But when this diet, normal diet, is modified with adding vitamin B12 or folic acid, choline or vitamin, you can find the lean mouse and obesity is less and they are not prone to the disease. So I have taken this is from a book. These are the from a book. This is the citations reference. So this is also I have taken the same things here they represent in NIEHS, National Institute of Environmental Health Science, USA. This is the examples how this environment or diet can influence your epigenetics. So now the question is how this happens. Then what is the mechanism behind this? One example, this is the case is called agouti viable yellow genes. This gene is only found in mouse, not in human. Though human and mouse both are mammals and they have a very close genetical relationship. But here you can see this particular gene only found in the mouse. This AB locus, okay, this is N transposons. This AB locus is actually retrotransposon, that is jumping gene that is inserted upstream of the agouti genes. Okay, this is the agouti mouse. They are called. This particular gene is present in the agouti, and they are present in a means number of copies present in the mouse genome. But when happen, these AB elements are methylated. Okay, this particular transposon is methylated, and they are normally shut off. They are not turned on. They are shut off or turned off. However, when agouti mouse, they are unmethylated. When they take some diets, this methylated locus is unmethylated, and when it is unmethylated, the promoter is active, and they leading to the yellow coat. The gene express leading to the yellow coat, and they are highly obese mouse as well as they are prone to different diseases. These environmental effects as well as diet you can also observe in other parts. Here, one example is in human condition, how the psychological stress can influence. This is one example is children who were conceived during a harsh wartime famine in the Netherlands in the 1940s. Here you can see they have the more diabetes, heart disease, and other conditions in Netherlands in 1940s. And this study already published. These are all are available in the online. So it means in other conditions, you can also means if you compare means two families, means suppose one mother is under stress condition, he deliver a child, and one in normal condition, you can observe the psychological difference of the both children. So this is the summarize of that. This is the latest one. Means one of this beautifully I have taken. It is from the Frontier Cell and Developmental Biology, published in 2014. Here you can see these are the all things already I have discussed, shown it. Psychological state, social interaction, your epigenetic composition is completely dependent on all these things. Even your friend circle, social interactions. If you have a bad friends and if you have a good friends, your epigenetic composition will be different from those who are interacting with the good people and those who are interacting with the bad people. They have the epigenetic composition is totally different, and these are all published data. Okay. So your social interaction is also depends, alternative medicine, therapeutic drugs, microbiome, exercise or physical activities, financial status, because you're depending on financial status, maybe you are under psychological pressure or economic pressure. This is the drugs of abuse, toxic chemicals, disease exposure, diurnal seasonal correlations, etc. So now Before going to the precision part, we should know the historical background of this epigenetics. Okay, so if, because this is a this is though it means complete work on epigenetics started after 1951 or 1942, but though 1942 it was discovered, but the work has been started after 1957 or 51, you can say. So in 1751, first the theory of epigenetics was given by C.F. Orr. And according to this theory, you have started this theory in my evolutionary biology book. Structures that have not yet developed emerge throughout the development. Okay. Throughout the development, what does it mean? It means your genetics are same. You inherited from your parents. Genetics is same, but it is developed because those organs which are pre-developed, they are developed because of the changes of your gene. And these changes is not the nucleotide composition. The changes in the epigenetic changes. But at that time, it was not discovered what is epigenetics or something else. So now 1802, Lamarck proposed the environment might directly or relatively alter phenotype, and you know this is the his most famous theory, use and disuse, and this one is also one of the famous theory by J.P. Lamarck. He was a famous French evolutionary biologist. Then in 1879, Fleming coined the term chromatin to refer to the structure of the stainable cell nucleus. The 1898 discovery of nucleotide by Raphael, known as tuberculinic acid, which is not recognized as a prerequisite or identification of DNA methylation at that time. In 1925, Johnson and Coghill reported detecting a minor amount of methylated cytosine, and this is the first identification of epigenetic changes of a nucleotide. Now, in 1942, the term epigenetics was coined by Waddington of Cambridge University. 48 discovers first naturally modified DNA nucleotide, that is the cytosine, in a chromatography of calf thymus DNA. 51 wet first the isolation of 5 methylcytosine, then create a model of epigenetic landscape. The same Waddington who coined the term epigenetics. Then size 61 chromosome inactivation we have by Leon hypothesis, you know it. Then 62 Arbar and Lin discovered histone methylation, 64 histone acetylation, then 65 DNA methylation. First structural resolution of nucleosome it was in 1974. In 1975, DNA methylation proposed as a process for the embryonic silence of X chromosomes. This idea that DNA methylation may regulate gene expression was proposed by Holliday and Ricks. 77, discovery of histone ubiquitination. First proof that DNA methylation is responsible for X chromosome silencing. So we know this. It was though it was discovered that this bar body or X chromosome silencing means we have studied in genetics. It was discovered earlier, but first time it was proposed that or proved that DNA methylation is responsible for X chromosome silencing. Then bromodomain or prions in 82, 85 to several divide acycline or cytotoxic agent developed by Upjohn inhibits DNA methylations. Then some drugs were discovered around 1982. Then this widespread loss of DNA methylation found on cytosine guanine island in a tumor samples. And from onwards, it was discovered in Johns Hopkins University, USA. And 1982 onwards, this epigenetics was related to the cancer. So there are so many people started to explore the possibility of epigenetic changes and how they are related to the cancer. Not only cancer, other disease also. So this is 85. Again, DNA methylation takes place on particular DNA regions known as CPG islands. Then cloning of this DNA MT or DNA methyltransferase genes. Then suggested inactive tumor suppressor genes. DNA methylation suggested in 89. They first proposed that DNA methylation is responsible for the tumor suppressor genes. We know there are two types of genes found actively involved in cancer. One is oncogenes and another one tumor suppressor genes. So tumor suppression, they suppress the genes, and oncogenes, they promote the genes, cancer. So oncogenes promote the cancer and tumor suppressors suppress the cancer. Then subsequently, there are so many discoveries you can see. The first conclusive study to show that decreased methylation of DNA resulted in the onset of cancers. And this methylation specifically, they decrease on the tumor suppressor genes. Sorry, oncogenes. Now in 1996, discovery of histone acetyl transferase and histone deacetylase, discovery of RNA interference, and first 1999 first demonstration in mammals is that epigenetic alternation may propagate through the generation from one generation to another generation, and then also linked with the colorectal cancer. In 1999, first time it was reported that. Then in 2001, chromodomain obtaining protein was discovered. Then 2004, identification of FDA approved and DNA methylation drugs first in 2004. Then 2006, another drugs. And I have shown you there are so many drugs are online. They are either in clinical trial 1, 2, 3, 4, or some are somewhere in the clinics. Now the next question is, what is epigenome? We have started the epigenetics. Now the next question is, what is epigenome? The epigenome is a multitude of chemical compounds that can tell the genome what to do. Means it includes, epigenome includes not only the methylation, it includes the acetylation, ubiquitination, somatylations, acetylations, or phosphorylation, etc. The human genome is the assembly of DNA. We know it. Almost there are approximately 3 billion base pairs are there in the genome, and this epigenome is made up of chemical compounds and proteins that are attached to DNA and direct the action of genes, whether it should be turned on or turned off, or shut on or shut off, controlling the gene expression in particular cells. Some examples are there. Suppose we have the same DNA both in hepatocytes, in islets of Langerhans or gamma cells, beta cells, skin cells, mouth cells, blood cells, WBC, RBC, every not RBC or WBCs, and other cells also, maybe in brain cells or neuronal cells. We have the same DNA, same person, they have the same DNA. Now, how the cells know? Means where already in the video you have seen in the liver, what happens? Or in the liver, in hepatocytes, some genes are expressing. In the muscle, some genes are expressing, but not the other genes. Suppose in insulin gene is expressing only in the pancreas, not other organ. So why? What is the reason? Because they have the genes, they have the gene sequences. So these controlling or differential gene expression is largely controlled by this epigenome. Now, what makes up the epigenome and what does epigenome do? The first is what makes up the epigenome. The epigenome is the set of chemical modification to the DNA and DNA associated proteins. What is here? This DNA associated proteins, maybe the histone proteins, which alter gene expression, and they are sometimes heritable through meiosis or mitosis. Now, the modification occur as a natural process of development or tissue differentiations, can be altered in response to the, I have already told you, different reasons, maybe diet, environmental exposure, disease, or microbiota. We have trillions of cells specialized for different function in muscles, bone, etc. Each of these cells carries the same genome. The epigenome controls many of these genes to the genome. In this way, cell can remember which genes are turned on and turned off, when, where, and how. Now, these are the summarize of the epigenetic changes we observed. This is, I have taken, this is from one of my recently publications paper. So these are the DNA methylations, histone modifications. These are the some examples. And this histone modification, then there is a microRNA or long non-coding RNA, specifically you can say non-coding RNA. They are also involved because this non-coding RNA include this here. I have mentioned only microRNA, but some non-coding RNA, they are directly involved with the, suppose, one example, very good example is X chromosome inactivation. And this X chromosome inactivation is, this chromosome is inactivated by a long non-coding RNA. This is called cyst. They wrap the whole chromosomes and the chromosome is unable to express, okay, throughout the life. Then another one is, this is the RNA methylation. This is a new field. And this recently we observed that maybe in 2010 or 2011, something like this. This N6 methylation of the adenine residue of mRNA, these are epigenetic changes in RNA, and they are directly related to the gene expression as well as in cancer biology. There are so many people are working with this, specifically some of these enzymes which involved in adenine methylations. They are MET L314, MET L14, F16. There are means you can just make a search on PubMed. You will find there are so many papers are coming. This is a very hot field nowadays in cancer biology. Now, this major multiplication here, you can see this. I have already discussed. This is the methylation, acetylation, ubiquitinations, and this is a not a very recent data. It was published in 2016 in Nature Reviews. Here you can see this is the methylations means in the histone, which histones are methylated where, and what is the effect of this methylation. Sometimes methylation not always repress or suppress the genes. Sometimes they activate the genes also. And here one example is H3 histone 3K4. Here you can see this is the acetylation, some of the, but another means in general, we understand that methylation causes gene suppression, acetylation causes gene expressions or activation. But this is not always true. Here you can see sometimes the acetylation also causes repression. And this is the ubiquitination. Now, the histone molecules, we know, we already discussed the DNA. Means we have the main target is the CPG, and sometimes also in adenine, guanine also. But in the most epigenetic changes, what we observed, this is mostly in the histone tails. And these histone tails here you can see, this is the H2A, H2B, H3, and H4. We, as already you observed in your video, there are 4 tetramer of this histone proteins, and they form the nucleosomes, and the DNA wrapped around the nucleosomes, and they compact. Finally, they form chromatin, then chromatin to the chromosomes, and chromosomes, how they are compact. We observed what we observed during the mitosis or in under the microscope. So here you can see these tails, H2A, these are the tail regions of H2A, H2B, H3, and H4. And these are the different types of modification we observed in the tail part. Here you can see phosphorylation, methylation, acetylation, or ubiquitinations. Now, these modified tails, then they recognized by some domain containing proteins, and these domain again divided into the 4 parts. They are bromodomains, chromodomains, pseudodomains, and PSD domains. And these proteins, when bind to the modified part of the histones, what does it mean? These bromodomains containing proteins interact only the acetylated histone tails. Then chromodomain or pseudodomain proteins contains only interact with the methylated histone tails. Means histone tails, here you can see this is the, I am talking about this one. When it is a methylated or when it is an acetylated, a particular type of protein comes and bind to the tails, and where these proteins, they have some domains, and this domain interact with this modified part of the histone tails. Then another protein scent domain containing proteins interact preferentially with unmodified histone proteins or histone tails. So basically, remember just 2 domains containing proteins. One is bromodomain, one is chromodomain. And this PSD domain, they are generally observed in the plant. But it has been reported recently in the animals too. Fingers specifically recognize modified forms of histone tails. Now, in many instances, remember these things. It is not always, if you have the modified acetylations or methylations, this domain containing protein binds always with all the modified pattern. No, it has been observed that one protein, example HP1, this is a chromodomain containing protein, that will bind only to the methylated lysine 9 of H3, only methylated lysine 9 of the H3, but not to any other site of the histone methylations. So some are highly specific to the some sites, and depending on the developmental condition, depending on the disease conditions, the situation is observed in different proteins with this domain containing proteins. There are proteins also reported that, suppose there are proteins that contain PSG finger specific for methylated lysine of pH3 immediately next to a bromodomain capable of recognizing an acetylated lysine. So there are 2 proteins can bind in the same sites. Now here you can see, suppose this is an histone octamers, and this is here you can see these are the tails, histone H2A tails, H3A tails, H2B, H4, and H3. And this picture is, you can found it in the Watson, J.D. Watson books, the molecular biology of the gene. I have taken this picture from that particular book. So this is here you can see this is the unmodified conditions, and maybe sometimes this unmodified histone core maybe methylated or maybe they are acylated depending on the developmental condition or disease conditions. Now, when they are acylated, bromodomain containing protein comes and they bind to the particular regions. Here you can see this is the modified acetylated sites. They bind, and according to the need of the cells, mostly they turn on the gene. Here you can see methylated one. You can observe they are comes chromodomain containing proteins comes bind here. And one beautiful examples of bromodomain containing the transcription factors. They generally bind when this bromodomain, these factors bind to the, when this tail is acetylated, these transcription factors comes bind and they transcribe the gene. So these, most of the transcription factors, they have the bromodomain. Now here you can see this is the chromodomain, and this chromodomain bind, they methylated and they suppress the genes. Now the question, how do the domains that recognize modified histones alter the function of the associated nucleosomes? So one important way is this modified histone recruit enzymes that will further modify the adjacent nucleosomes. Examples, many of the enzymes that acetylate histone tails, this is histone acetyl transferase or heads, include bromodomain that recognize the same histone modification that they create. Okay, that they create. Even this bromodomain or chromodomain, they have the similar functions, and sometimes this particular domain, this modification also can inherit from parents. Now, this is the transcription factors. I have already told that this chromodomain containing or methylated containing domains mostly observed in the polychrome group of proteins. You know, they are directly involved in cancers like BMI1 or EGS2. They are directly involved in most of these cancers, and their expression directly related to the methylation of the most of the tumor suppressor genes, and subsequently they promote the cancer. Now, the histone modifications we have just described are dynamic. They are catalyzed by specific some enzymes. Mostly remember this, very few are inherited. Very few modifications, epigenetic modifications are stable. But most of the epigenetic changes, they are dynamic and they are catalyzed by histone acetyltransferase, histone deacetylase. If from the name you can understand what is the function of this particular enzymes. HEADs, HDX or HMTs, methyltransferase or demethylases. One enzyme add the acetyl groups, another one remove the acetyl group, one enzyme add the methyl group, or they remove the methyl group. So these are the readers, eraser. These are the one function, another function of these enzymes. And currently here you can see this is the summarization. This is also from the latest Watson book. Here you can see what are these type of enzymes. Histone acetyltransferase complex or deacetylase complex we have already known. And most of the drugs we design again the epigenetic changes. We target this particular group of proteins. Here you can see first one is acetylase transferase, deacetylase transferase, methyl transferase, or demethylases. And the detailed function is also there. If you want to study in details, you can refer the Watson book or any other good molecular biology book. You can find this latest published book, means maybe 2023 or 24 after publication date. Now, this identification of acetyl, this is one example. I have not studied it. Please don't take the pictures. Recently, we identified one new in histone 2A, one acetylate amino acids, and we are going to publish it soon. So this is just a technique how we identified it, but through MSMS. Now, is the epigenetic is the epigenome inherited? I have, as I have told you, some epigenetic changes, they are inherited. The genome is passed from parents to their offsprings and from cells when they divide to their next generation. Much of the epigenome is, as I told you, they are dynamics. They are dynamic. They are some part of the life you can observe it, some part of life you don't observe the expression. Much of the epigenome is reset when parents pass their genomes to their offspring. However, under some circumstances, some of the chemical effects on the DNA and histone of eggs and sperm may be passed on to the next generation. Now, when cells divide, often much of the epigenome is passed on to the next generation of cells, helping the cells remain specialized. Can the epigenome change? Although the cells in the body contain essentially the same genome, the marked by chemical text on the DNA and histone gets rearranged when cells become specialized. Now, if you want to study in details about this, how this you can I can suggest you one book. This is the Albert book, and how this cell reprogramming happens. Cell reprogramming means they explain it in details. So for this, can the epigenome change? You can found in details about these things in the Albert books. Now, what makes the epigenome change? As I told in one previous figures, there are maybe smoking, diet, infection, disease, etc. Now, another question is, now there are two things. Another thing is imprinting. So whether the imprinting or epigenetics is same, no. Imprinting is a type of genome imprinting is a form of epigenetics. Okay, not all epigenetic changes are imprinting. So imprinting is just a form of epigenetics. It's a part of epigenome. Now here you can see this when this imprinting or this epigenetic changes, they are or means this wrongly controlled or wrongly regulated, they cause different disease. Here some examples are Beckwith-Wiedemann syndrome, a disorder associated with body overgrowth and increased risk of cancer. So this imprinting, though this imprinting is observed in a normal person also, this is a normal process. Another one is Prader and Willi syndrome associated with poor muscle tone and constant hunger leading to obesity. Then Angelman syndrome, which leads to intellectual disability as well as motion difficulties. So these are the some of the things which this disease caused due to the abnormal regulation of the imprinting. Now the question is how this imprinting control the genes, though it is a part of epigenetics. It is a form of epigenetics. I am citing here only two examples. One is IGF2. Another gene is H9. H9 is a long non-coding RNA, and IGF2 is what is called insulin growth factor 2. Now, this we know means in most of the mammals or eukaryotes, they have the what is called homozygotes or heterozygotes. They have the duplicate genes, two genes in each allele. Or sorry, each gene has a two allele, and one allele from the mother, another one allele is from the father. So here you can see, in some cases, what happens, sometimes this imprinting means this expression of gene depending on which parents the gene is inherited, if whether it is from the maternal side or whether it is from the paternal side. So sometimes some genes, they are imprinted or they are silenced or they are activated depending on the which parents the gene has been inherited. So here you can see this is two examples, beautiful examples. IGF2, insulin growth factor 2 gene, and this is long non-coding RNA H9 So here you can see CTCF or CTCT factors which bind here. Okay, what is this? Is the imprinting control regions. Here you can see these imprinting control regions are present in both the genes. This is P means this is the paternal, and M means this is the maternal side. Okay, so there here you can see this is the two genes. One is one allele is from maternal side, another allele is from the paternal side. Again, H19 also from the maternal side and from the paternal side. Now, first we observe the regulation of H19. When it is from the maternal side, what happened? When this H19 is from the maternal side, CTACT factor bind to the ICR regions. Okay, this is the ICR region. This is the imprinting control region or ICR region. ICR imprinting control regions. So what happened? This CTCF bind to the imprinting control regions and they prevent the methylations. Okay, they prevent the methylation. So when the DNA is not methylated, this enhance the expression of this H19 gene if they are inherited from the maternal side. But what happens when they are inherited from the paternal side, same gene, okay, from the same allele, when it is from the paternal side, they are unable to express why there is no, this gene is methylated and there is no CTCF factor. So this gene is unable to express if they are inherited from the paternal side. Same in the IGF2, they are from the, if they are the maternal sides, they are unable to express. But if they are from the paternal sides, what happens? There are two genes, DMR1 and DMR2. So this DMR1 and DMR2, they are directly involved in the gene expression and suppression. When this ICR region, imprinting control regions, is methylated by DMR1, the gene is suppressed. But when these are methylated with the DMR2, the gene is expressed. So what happens? This if these genes are from the H9 is from the paternal side, they are suppressed. But what happens? This insulin growth factor 2 or IGF2, they are from the paternal side. These DMR2 bind and they express or enhance the gene expression of the IGF2. If it is the but in case in maternal side, they are the suppressed, they are unable to express. So this how this IGFR2, this there are so many conditions depending on this implanting. If they are normally regulated, no problem. But if they are abnormally regulated, they cause IGF2 cause different types of disease, from cancer to the neurological to the growth and metabolic disorders. Like this is H19 also cause different type of disease, cardiovascular, aging, oncogenic, and so many things. Now, here you can see now the formation of a cancer cells by epigenetic changes, how they form. If this changes, this is from the AFCOM websites. Here you can see this is the normal cells. Then epigenetic changes, but these epigenetic changes may cause the DNA alterations. So this cancer is initiated, or these changes can cause this is the epigenetic changes, this methylation, suppression, or something like this. They also and finally the cancer initiated also DNA alterations, and finally these cancer initiation, cancer progression, metastasis, DNA changes, all these causes the cancers. So some of these examples are reported from the journals. I have taken from this, how DNA methylation inhibit the tumor suppressor genes, or methylation. Also, this pattern is varies from tumor to tumor too. So this is how this is the expression. This picture is from the Nature reviews. So this I summarize this from the Lancet Oncology, published in 2002. How this hypermethylation observed in some of these tumor suppressor genes, and hypomethylations observed in the oncogenes. Okay, so this is how this observed. They are summarizing cell control, invasion, and imprinting, aging, these factors, environmental, hormone, genetics. They are summarized, and I took this picture is from the Lancet Oncology. Now this now the question is food. I have one of my colleagues, he is working on this particular field, how this food modified this epigenetic and cancers. And here you can see some of the things. This is the cruciferous vegetables and fruits. They contain the quercetin and resveratrol. There's curcumin from turmeric, green tea, GCG, and ginsetin from the soybean. How they contains or they influence the means cancers, as well as how they are nowadays taking as an adjuvant therapy or neoadjuvant therapy or monotherapy against different cancers. The most important thing is that when this epigenome is changes abnormally, they developed either this particular cell is radio resistance or chemo resistance. Chemo resistance means they are drug resistant. Radio resistance means they are resistant to the radiotherapy. And when you take this I will show you one case study recently published in 2022 in my last few slides. So this, when you are taking with the adjuvant therapy or neoadjuvant or monotherapy, they can chemosensitize or radio sensitize to the cancer or tumor cells. And some of these things here you can see already in published data, they are available. This is in chemotherapy and how they work as an adjuvant therapy. So DNA damaging agents like cyclophosphamide and other drugs, and this radiotherapy, how they work together with some bioactive phytochemicals. They inhibit the DNA methyltransferase. Some are histone deacetylase inhibitors, some are microRNA inhibitors. By different routes, they work. These phytochemicals, active phytochemicals, they work, and they chemosensitize or sensitize or prevent the cancers. Now this contribute. This is summarized. I am summarizing it. How do changes in the epigenome contribute to cancer? Changes in the epigenome can switch genes involved in cell growth or immune response. These changes can lead to the uncontrolled growth or hallmark of cancer, or to a failure of the immune system to destroy tumors. So this one example is this is the glioblastoma. Here you can see patient treated with the temozolomide, okay, which kill cancer cells by adding methyl groups to the DNA. When it is methylated, they can preferentially kill the glioblastoma cells, cancer cell. So these are some other examples also there. Now, epigenetics, a personalized approach to cancer therapy. As in our first 2-3 slides, we discussed these epigenetic changes can vary even from the monozygotic twins. Monozygotic twins, they have 100% same genetics, but there are some different characteristics due to the epigenetics. So, this from these particular examples, we can refer that epigenetic changes are patient-specific, leading to a personalized epigenetic therapeutic approach. Now, this epigenetic profile of each patient, they are treated with a specific dose of the drug. We can make a profile, and then what happen in each individual according to the based on their epigenome of each patient, we can give them the inhibitors, whether it's the DNMT inhibitors or SDEC inhibitors or HET inhibitors. Okay. Now, here you can see this particular region in conventional treatment. Here you can see this is also from one of my students' paper. She is now in USA. So means here you can see in conventional treatment we give the response. So out of these 3 patients, here you can see sometimes all are no response. Some have adverse effects. A few populations they give the response of the drugs. Some population they don't have the response, and some population they have the adverse effects. But when we precisely design the medicines, maybe against the epigenetics or sometimes in genetics also, you can see we may observe the response against all these patients. Okay, those who are not respond to the conventional therapy, they also respond. Now, so this personalized medicine, here you can see this treatment testing, general treatment, then this clinical biological correlation, and this is the omics profile. Based on this omics profile, we can design the medicine in each individuals. Now, one case study, as I told you, I will show you one case study how these epigenetic medicines work. Okay. One case study here you can see this is a rare presentation of non-Asian. It has been published in 2022. Non-Asian female with this is a study in USA. That's why non-Asian. Non-Asian means she is an African origin. Okay. So with metastatic non-small cell lung cancer harboring EGFR L747P mutations with clinical response to multi-targeted epigenetic and EGFR inhibitor. Okay, or inhibition. So this is the first in 2013 August. She reported diagnosed with different conventional medicines. Medications are there. Here you can see this is the osimertinib, then ipatinib, and this is erlotinib of the combined epigenetic treatment. They for the combined they are selected a mixture of flavonoid and phenyl butyrate. These are the epigenetic drugs. Okay. So this EP drugs, you can say the EP drugs. Though, so the epigenetic treatment is introduced by intravenous injections. Now, when they combine with this EGFR inhibitor with the epigenetic treatment, from November 13 to 14, patient received several therapies, which included 4 rounds of stereotactic radiotherapy, the paraspinal tumor, radiotherapy along with the right medial lobectomy. Lobectomy means the cut off or removal of an lungs loop for the adenocarcinoma, which was in stage 1. Then chemotherapy cycles with Alimeta and carboplatinum. A laboratory test confirmed the presence of spreading cancerous cells along with the mRNA of the oncogenic protein MYC and the chromatin modifiers HDEC. So immediately they started the epigenetic chemotherapy administered 5 times weekly. The diet plan remained unchanged for that particular patient. In 2015, the researchers lowered the erlotinib dose 25 to 50 grams per day to avoid combination side effects with the epigenetic treatment. In 2015, July 2015, cutoff from epigenetic therapy occurred because the patient could not take further epigenetic therapy, which caused a disease recurrence. Then in July 18, erlotinib resistance was found. Osimertinib has been given for 3 years. Unfortunately, some epigenetic drugs showed some side effects, so an epigenetic switch has been done. Now here you can see in July 2020, a localized radiation was done on the pulmonary lesions. A disease progression was again found during that time. Osimertinib at the dose of 80 milligram per day was going on. After the laboratory test, the epigenetic protocol was placed for the patient, and on the treatment with afitinib, the patient decided to reduce the epigenetic therapy to once per month, which causes disease increasement. Now, June 21, after molecular analysis, advancement of gene mutations have been found in TP53, AKT, and retinoblastoma. Therefore, they were restarted the epigenetic therapy combined with decomitinib of 45 milligram daily on a 5-day on-off schedule. Epigenetic therapy was continued 4 times per week for 6 weeks. The treatment protocol was completed on April 23, 2021, and laboratory investigation were conducted on 15 June 2021. The summarization of the result is, in this study, epigenetic therapy combined with the low-dose treatment targeting EGFR has demonstrated a very good progression-free survival, with more than 8 years of life expectancy and no severe or chronic side effects. So here you can see a 56-year-old non-Asian woman with oligometastatic NSCLC. Before study, various types of treatments were there, as we discussed. Based on the biomarker detection, patient was treated by epigenetic therapy. Now, then treated the patient with EGFR inhibitor plus epigenetic therapy. Treatment only with in a few months. Treatment because of the side effects. The treatment only with the EGFR inhibitors. Again, treated patient with epigenetic therapy and plus EGFR inhibitors. And this one is here you can see 8 years survival without suffering from any serious side effects. So we don't know whether she is alive now or not. So this was published in 2022, and now 2025. If she is alive, maybe it means more than 8 years. Okay, 8-4 means almost 11 or 12 years. Now the conclusion is, in recent decades, personalized epigenetics has been gaining more and more attention from the researchers for its fine application and impact on disease. In numerous clinical trials, epigenetic therapy has demonstrated a remarkable therapeutic result when treating patients with different forms of cancer. Accumulative evidence showing its importance of epigenetic therapy in the field of cancer. However, several combination therapy trials are needed for this personalized epigenetic treatment to make a firm base in cancer treatment. The mentioned case study showed that combination therapy with epigenetic treatment might be one of the choice for treating cancer patients. In the future, a greater number of clinical trials will be needed. It is important to carry on this type of combination therapy for better advancement in personalized epigenetic therapy. And these are the some of the citations. Thank you. Thank you very much for your attention.
Thank you, sir. Thank you very much for this informative session.
If you have any questions, you can ask.
So there are a few questions in the chat box. I can read it for you.
Where is chat box?
Hafsa Arif has asked, sir, what strategies can overcome poor bioavailability and pharmacokinetic limitations of epigenetic phytochemicals in clinical approaches?
Yeah, there are so many things are there. I mean, say this poor bioavailability not only problem with the phytochemicals. This poor bioavailability also we observed in some other synthetic drugs too. But scientists are working. This is one thing is that this nano medicines or nano delivery or formulation of nano particles of this particular drugs. So this is one of the solutions. Yeah.
So there is another question by Nilofer Banu. She asks, how do epigenetic modifications such as DNA methylation, histone modifications, and non-coding RNAs contribute to the development of cancer drug resistance? And how can these changes be therapeutically targeted?
So means, there are so many genes are there. Those genes are directly involved with the chemoresistance. Suppose polycomb group of protein. This is one polycomb group of protein. This is BMI1. So what happens when most of these oncogenes, what happens? They are acetylated. They are overexpressed. They confer the chemoresistance. So this is the epigenetic changes. Though the DNA sequence, all these oncogenes are not mutated always. So they confer, when they overexpress, they confer the chemo resistance. Okay. Similarly, in tumor resistance or tumor suppressing genes, they are, when methylated, they are suppressed. So they are unable to cope up with the tumor load. So by that way, these epigenetic changes can either promote or they can suppress the cancer. Is it clear, Hafsa? No, this is Nilufar Banu, I think so. Nilufar Banu, is it clear?
Yes, sir, she has responded with a thank you, sir. Their mic has been muted, so they are replying in the chat box.
Oh, okay.
There is
Okay.
another question by Khalida Siraj. She asks, well, will antioxidants present in epigenetic photochemicals not lower the effect of chemotherapy?
Antioxidant present in epigenetic phytochemicals. There is a beautiful articles I have published in 2023, whether these phytochemicals are foe or friend in antioxidant cycles. You can read it. This is publicly available. Okay, these are free accessible. Where we can example, so many examples I have. If Ms. Khalida, if you just give your mail ID to me, I will forward you this article. There are so many examples are there. Okay. So we have put whether they are, sometimes they are for these phytochemicals, sometimes they are friend. Not always these phytochemicals are anti-cancer activities. Sometimes some phytochemicals, they also promote the cancer activities. And we have listed a group of phytochemicals in that particular paper. It was published in 2023. So it is not very old. It is a very new article. I will forward to you. You can find the details, how they work, what is the mechanisms, how they promote the cancer, and sometimes how they inhibit the cancers.
Okay, thank you very much, sir, for a wonderful talk. Now it is time for a short break, and let's gather us all after lunch at 2 p.m. And sir, Khalida has shared her email address in the chat box. You can find it there.
Yeah, you can. I have also share my. If someone has some questions, they can ask me.
Okay. Okay. Thank you very much, sir.
Thank you very much.
Thank you, Dr. Hifzur. I would request you to please provide the slides for the participants to be accessible. So I'll upload it on the GAN website in the Google Drive that we have.
Okay, sure.
Thank you. Thank you so much. Thanks to all the participants who are here, and we'll see you at 2 p.m. with Dr. Hassan.
Hopefully, I am on time now. 1 hour.
Perfect.
Yeah, thank you very much for giving this opportunity.